Hello everyone, my name is Dhanushri, second year BE in Information Science and Engineering student at Sri Venkateshwara College of Engineering, Bengaluru. Today's topic of my presentation is DNA fingerprinting. Agenda, Introduction, what is DNA fingerprinting, how is it carried out, procedure, pros and cons, DNA databank system, applications, conclusion. So here you can see these two people are communicating about the DNA fingerprinting. Did you know that they created something called DNA fingerprinting? Then the person he reacts that no, I did not. What is it about? So then, well, DNA fingerprinting is when stuff from your body like blood or fingerprints is used to identify you. That's very interesting. Yes. DNA fingerprinting, I think a lot of people are introduced to DNA fingerprinting first while watching a crime shows. An officer collects some samples from the crime scene, they put it in a tube, and then an hour later they hold up a brightly colored gel, squint at it, and say, "Aha, we have a match for a killer's DNA." Of course, this isn't exactly how things work in a real life, but DNA fingerprinting is an important part of a forensic science. DNA fingerprinting is the laboratory technique used to determine the probable identity of a person based on nucleotide sequences of a certain regions of human DNA that are unique to individuals. DNA fingerprinting is used in a variety of situations such as criminal investigations, other forensic purposes, and paternity testing. In these situations, one aims to match two DNA fingerprints with one another, such as DNA sample from the known person and one from the unknown person. How is it carried out? Firstly, isolating the DNA, digesting the DNA with the help of restriction endonuclease enzymes, and then separating these digested fragments as per the fragment size by the process of electrophoresis, and then blotting the separated fragments into synthetic membranes like nylon, hybridizing the fragments using labeled VNTR probes, analyzing the hybrid fragments using autoradiography. Procedure. To get your DNA fingerprint, you'd give a sample of cells from your body. This can come from a swab inside your mouth, your skin, the roots of your hair, or your saliva, sweat, or other body fluids. Blood is usually the easiest way. Lab workers treat this sample with the chemicals to separate the DNA, which is then dissolved in a water. Your DNA is cut into a smaller segments with another chemical process to get sections of a 5 to 10 base pairs that repeat themselves. Technicians copy these tiny sections millions of times to make the samples longer for easier study. Lab workers take those strips of DNA and mix them into a gel, and then they run an electric current through the gel, which separates smaller strands of the DNA from the larger ones. A dye added to gel makes the DNA strips stand out when they are placed against ultraviolet light or lit up with a laser. The more these short segments are tested, the more accurate the DNA profile will be. The strips will show a barcode-like pattern that can be compared to the results from one another sample to find the match. So here you can see in this diagram, firstly we will collect the blood sample, DNA is then extracted from the blood cells, and then DNA is cut into fragments by a restriction enzyme. The DNA fragments are separated into bands using electrophoresis in an agarose gel. The DNA band pattern in the gel is transferred to a nylon membrane by a technique called as a southern bottling. So then the DNA probe binds to the specific DNA sequences on the membrane. Access DNA probe is washed out. At this stage, the radioactivity probe is mounted to DNA pattern on the membrane. X-ray film is placed next to the membrane to detect the radioactive pattern. The X-ray film is developed to make the visible pattern of the bands, which is known as the DNA fingerprint. Pros and cons of DNA fingerprinting. Pros of DNA fingerprinting are DNA fingerprinting is a non-invasive and painless medical technique to gather genetic data. DNA fingerprinting is useful for the wide variety of cases ranging from a crime investigation to evolutionary studies. DNA fingerprinting also helps solve parental disputes to determine the parental pattern in case of a denial. Then DNA fingerprinting is easily accessible from the body fluids or hair follicles. Cons of DNA fingerprinting are DNA fingerprinting can be missed due to its easy accessibility. So then performing the test without someone's prior knowledge is a violation of privacy. This information could be handled by third parties to wrongly convict an innocent individual. Does DNA bank system help solve crimes? Do you think so? Then the current DNA database maintained by FBI, known as the Combined DNA Index System (CODIS), that is CODIS, then contains say case samples, that is DNA samples from the crime scenes of or a rape kits, and individual samples collected from a convicted felons and or arrestees, and these are compared automatically by the system software as the new samples are entered. As of February 2007, Codis has produced over 45,400 hits, which assisted in more than 46300 investigations that is Federal Bureau of Investigation. However, contrary to how DNA analysis is portrayed on popular television shows, DNA samples are not analyzed with the course of an hand. Rather, the US currently has an enormous backlog of samples waiting to be typed and entered into the database. So some of these samples are from the cases that have outdated their statutes of limitation. So even if these samples could help solve a crime, the crime no longer be tried. So here is an image which says the police collects the traces of the body tissue from the scene of the crime, a smear of a blood or a skin or a scrap off in a struggle. So then in the laboratory, the forensic scientists extract the DNA from these cells and compares it to a DNA taken from a suspect. And then the third step is identical DNA links the suspect to the crime scene, and then the suspect is arrested. Applications of DNA fingerprinting, paternity and maternity. Because a person inherits his or her VNTRs from his parents, so VNTR patterns can be used to establish paternity and maternity. The patterns are so specific that a parental VNTR pattern can be reconstructed even if only the children's VNTR patterns are known. The more children produced, the more reliable the reconstruction. Parent-child BNTR pattern analysis has been used to solve standard father identification cases as well as more complicated cases of confirming legal nationality and in instance of adoption, biological parenthood, etc. So the second application is criminal identification and forensics. So DNA is isolated from the blood, hair cells, or other genetic evidence left at the scene of a crime and can be compared through VNTR patterns with the DNA of a criminal suspect to determine the guilt or innocence. Personal identification. The notion of using DNA fingerprints as a sort of a genetic barcode to identify individuals has been discussed, but this is not likely to happen anytime in a foreseeable future. The technology required to isolate, keep on file, and then analyze the millions of very specific VNTR patterns is both expensive and impractical. So social security numbers, picture ID, and other more mundane methods are much more likely to remain the prevalent ways to establish personal identification. Some of its applications include in forensic science, as I already said, it is used to identify the prospective criminal suspects to prove the maternity and establish familial ties, to identify and protect the commercial crop and livestock types, to figure out an organism's evolutionary history and the relationship between the different groupings of species. Lastly, conclusion. DNA evidence is easy to obtain because genetic material is found in all human cells save red blood cells. As a result, when we leave behind small biological bits of ourselves, these bits can be used to identify us and link us to the places we have been. With modern technology, the amount of DNA required for analysis can be obtained from even a minuscule biological sample, which allows police to match crime scene evidence with suspects. However, because the forensics is science largely rooted in probabilities, even a confirmed match does not supply concrete proof of guilt. In addition, DNA databases designed to simplify the process of connecting the past offenders to the recent scene crimes are fraught with the concerns involving the individual genetic rights as well as problems related to the delayed sample entry, both of which hinder the ultimate usefulness of these databases. As a result, Even though forensics is undeniably important to modern justice system, its personal ramifications and ethical questions are topics of continuing discussion with the scientific, law enforcement, and legal communities.